My name is Angelina Olivia with student last ID number 28.
Hello, my name is Wiji Kurniawati. My student ID number is 53.
My name Hanifah Eka Cahyanti and my last study number is 057.
Hello everyone, introducing myself, my name is Hani Kartika Puspaningrum with the last study number 50H.
My name is Mefta Aisah, my last student ID number 230.
And I'm Michaela Joyce Boutista, student from Tarlac Agricultural University from the Philippines, with a student number of 2018 and 101158.
Plant defenses. There are two defense components. The first is structural properties that act as physical barriers and inhibit pathogen entry into the plant. The second is biochemical reactions within cells that are toxic to pathogens or create conditions that inhibit pathogen development. The combination of defense properties is certainly different. The factors that influence it are: first is age of the plant, second is type of plant organ and tissue attack, the third nutrient status of the plant, and the fourth weather condition. Next is structural defense. The defense structure before a pathogen attacks includes: the first is the amount and quality of wax and cuticle on epidermal cells, and the second is the size, location, and shape of stomata and lenticels. Wax on the surface of leaves and fruits, fine and thick hairs on the surface of plants form a water repellent surface, prevents the formation of a water layer. Pathogens cannot be deposited and germinate or divide. Infection decreases. The second, the thickness and strength of the outer walls of the epidermal cells inhibit direct penetration. Fungi and bacteria can enter through the stomata. Example, rust disease in wheat can only enter when the stomata are open. Wheat with stomata that are slow to open during the day is more resistant because the germ tube and germinating spores have dried out when the stomata open. The last, stomata with small entrance holes and tall and large opening cells are more resistant to certain pathogens.
Tissue defense structure. A. Formation of cork layer. Phloem or cork is a protective tissue formed by phloem outward. Phloem itself is cambium tissue that form the periderm. Periderm function as a protector or epidermis substitute in secondary growth. The function of the cork against tissue defense are as follows. 1. The cork layer serves to protect plant from physical injury when the woody layer of the plant is injured. The cork layer is thick enough to protect transport tissue and other essential plant tissue. 2. Cork cells are impermeable to water and air, making various pathogens unable to pass through. Thus, cork cells can protect plant from infection by pathogens such as fungi and also disease-causing bacteria. B. Formation of the abscission layer. Abscission is the process of falling plant organs such as leaf, flowers, and fruit without leaving roots. In the abscission zone, two layers or cell regions can be distinguished. The first layer is the abscission layer, which will undergo structural change to facilitate the separation of the organ stalk with the stem. The second layer is the protective layer that is formed under the abscission layer and protect the exposed surface when the organ fall, so that it does not dry out or become infested with a disease. The protective layer is formed due to the synthesis of various compounds in the cell wall and the intercellular space.
Next, from tissue defense structure, C. Formation of telosa. Telosa is protopalms that grows beyond normal from parenchyma cells, protruding into the xylem through openings. Telosa formed in the xylem due to stress conditions during vascular pathogen attacks. The large size and numerous quantities will completely clog the vessels, inhibiting the development of pathogen. Next, D. Gum deposition or blendok. Various types of gum are produced around the wound after infection by pathogen or as a result of damage. The gum quickly deposits into the intercellular space and within the cells surrounding the site of infection. Unimpenetrable barriers from pathogens become isolated, starve, and die. Next, cellular defense structure will involve morphological change in the cell wall of cell attacked by pathogen. There is 3 types of cellular defense structure. Number 1, swelling in the outermost layer of the cell wall that contact incompatible bacteria, preventing the bacteria from reproducing. Number 2, the cell wall thickness and response to pathogens, increasing resistance to penetration. Number 3, callose deposits on the inner side of the cell wall in response to fungi. Papillae are produced immediately after wounding. Papillae serve to repair cellular damage and prevent pathogen from penetrating neck-boring cells. It can form callose that wraps around the tips and of hyphae and forms a shield or tube of lignin.
Cytoplasmic differentiation. Slow-growing pathogenic fungi from hyphal clumps surrounded by cytoplasm. They become granular and hard. The pathogenic mycelium breaks down and attacks clumps. Cytoplasmic differentiation. Plant resistant by a substance produced by cell before or after infection that help the plant's defense properties. Necrotic differentiation affects through hypersensitivity, especially in disease caused by obligate parasite virus and nematodes. The necrotic tissues isolate the obligate parasite from the surrounding living substance. The pathogens depends on the food materials. The pathogens dies. The sooner the host cell dies after infection, the more resistant the plant is.
Metabolic resistance induced by pathogen attack can be among others, namely the first, biochemical inhibitors produced by plants in response to pathogen damage. The second is defense, third, hypersensitive reaction, and the third is defense through increased levels of phenolic compounds. In the first, biochemical inhibitors produced by plants in response to pathogen damage is plant cells and tissues reaction to damage by pathogens. The reaction is aimed to insulating the disturbance and healing the wound. Several types of compounds are produced in concentration high enough to inhibit the growth of fungi and bacteria. And the second, defense through hypersensitive reaction. The first is in resistant varieties. After infection, the cells will quickly lose their curtain bond and die. Change of occur in cells death and collapse in infected cells include The first is loss of cell membrane permeability. The second is increased respiration. And the third is phytoalexin production. Next, fungi and bacteria inside the cells where hypersensitive reaction occur are isolated by necrotic tissue and die quickly. Third, increased levels of phenolic compounds. Phenolic compounds are present in both healthy and diseased plants, but accumulation is accelerated after infection. Other phenolic compounds that are not present in healthy plants but are produced after being stimulated by the pathogens, phytoalexin in hypersensitive tissue.
Chemical defense before pathogen attacks, inhibitors released by the plants into their environment. And when the plant secretes a various substances through their surfaces that have inhibitory power against the certain pathogens, well, the example for this is the fungi toxic exudates on leaves can inhibit the germination of fungal spores. And when the plants have a way to defend themselves against pathogens such as fungi, bacteria, and viruses even before the attack, they do this releasing chemical inhibitors into their environment through their surfaces such as leaf, stem, and roots. Well, the chemical defense before a pathogen attacks defense in the absence of essential factors when the cell surface does have not specific recognition, the factors that can be recognized by the pathogen. So therefore, it does not attack the plant or the pathogen does not also produce infectious substances like for example the enzyme, the apresorium, infection tube, or the what we call hostorium. And these substances are in the form of oligosaccharides and polysaccharides, and also the glycoproteins or what we call lectins. Also, this chemical defense mechanism used by plants to prevent pathogens from attacking them. Receptor deficiency and host sensitive parts to toxin. If the specific toxin stick to sensitive parts or certain receptors in cells, well, the plants become sick and has symptoms. If the plant does not have these receptors, it remains resistant to the toxin and no symptoms. So the toxin produced by the pathogens can bind to specific receptor on plants, causing harm and disease symptoms. If a plant lacks of these receptors, it is resistant to the toxin and cannot be infected. The presence of receptor acts as a key for the toxin to gain entry and cause disease, while the absence of receptor keeps the toxin from causing harm, making the plant resistant. So the case study is the relationship of leaf tissue characteristic with the leaf attack on leaf blast disease, what we call in the scientific term of Pericularia oryzae cab onso dipadi oryza sativa L of genotypes. And this study came from the Ina Mustica et al., which is in the Department of Agrotechnology Study Program, the Department of Plant, Pet, and Diseases in Universitas of Indonesia. Well, the blast disease, which can reduce crop yields and by up to 70%, it infects all the growth stages of rice plants, namely leaves, books, panicle necks, and barely attacks the leaf midrib. Also, the Pericularia oryzae fungus that causes blast disease, it spreads through the air, attaches to the leaves through the water splashes, and then infects the leaves and causes the spot on the leaves. And this study aims to evaluate the resistance to leaf blast disease by looking at the one of the structural traits that are mainline of defense of plants it against the pathogen attack. It namely the thickness of the the leaf membranes or the epidermis, because it assumes that the thickness of the leaf epidermis of each rice genotype will be different, that will provide a good defense against the blast disease. It differentiates the responses to blast attack. Well, the conclusion that the structural defense characteristic of the leaves are the amount of the quality of the wax and cuticle covering the epidermal cells, while the structures of the epidermal cells, the size, location, and of course the shape of the stomata, the lenticels and the thick cells, networks that inhibits the advance of the pathogens. So this epidermal thickness is not, or is the one of the structural defense found in the plants even before the pathogens. Also, it comes from the contact with plants. So this thickness and strength of the outer wall of the epidermal cells is an important factor in the resistance of some plants and species. And to a certain pathogens, the epidermal cells with a strong and thick walls, it will direct the penetration of the pathogenic, fungi difficult, or even possible. Thank you for listening to our group presentation, that as we look to the future, we can expect that the continued advances in this field will lead to a new innovative approaches to plant disease management, and ultimately benefiting not only the agriculture but also human health and environment. Thank you.